PharmaVentures, experts in deals and alliances.
Today there's a great deal of interest in immuno-oncology as a method for tackling cancer, and there are several companies with lots of approaches. Typically people have attacked PD-1, PD-L1 and CAR-T. But how are these companies differentiating themselves? Are they all going after the same thing or not? With me today is George Rawadi of Celyad. He is the VP of Business Development and IP, and George's company is taking a slightly different approach, although it is CAR-T. George, welcome.
Hi Adrian, how are you?
I'm good, thanks. So thank you for joining us today. And perhaps we could start with a little bit about your company and the technology that you're employing in the fight against cancer.
So Celyad, it's a cell therapy company. We are based in Belgium, and we have offices and facility in Belgium and Boston, Massachusetts. The company since 2015 is developing a quite unique CAR-T assets. And why we say CAR-T unique in terms of we are leveraging the success of CAR-T in clinical trial, especially with the CD19, but we are not developing another CD19 CAR-T. What we are developing is totally a new approach of CAR-T using the biology of NK receptors. Those receptors are expressed naturally on NK cells, where they help to identify specifically tumor cells and activate NK cells to eliminate those tumor cells. So what we do, we take those NK receptors, we make them chimerics without any changes in their capacity of binding their ligands. Mm-hmm. And we overexpress them in T cell, and this is how we make our CAR-T. So this is the first time ever this kind of receptors are used in a CAR-T context, and this is what creates the uniqueness and the particularity of our approach.
And is this an autologous approach, or is it?
Well, I mean, today we are developing as an autologous approach. Obviously, we are also working on an allogeneic platform. Mm-hmm. So the same product, when it has a proven safety and efficacy, can be turned into a second generation allogeneic. So today autologous is the most straightforward, and I would say is the most easy way to prove concepts. I mean, we'll see how the business model around autologous is viable, but potentially all those compounds or all those cell therapies can be transformed into allogeneic later on.
So you mentioned there the traditional, if we can use traditional immuno-oncology approach, because it is very new, is to use CD19 as the mechanism for a CAR-T approach, and has been practiced with considerable success. I believe people are getting better than 80% response rates. Why have you taken a slightly different approach? Is it IP driven, or is it just because you have a better way of doing it?
Well, I think CD19 has generated great results, and this is creating real alternatives for CLL and ALL patients, definitely. However, CD19 is applicable to a limited number of what we call blood tumors or liquid tumors. There is a huge unmet medical need in many other cancer indications, such as prostate, breast, ovarian, pancreatic, and other type of liquid tumor where CD19 is not applicable. So that's why there is a great need of other target and other CAR to address those specific indication, and that has a huge number of patients that are underserved because most of them, they are running out of options. So what we do with our CAR-T is, because we are using receptors that doesn't target a specific tumor antigen, but they target a set of ligands that are expressed on most tumors. Today, data from Celyad and also from literature shows that 80% of tumor cells express at least one ligand that can bind to our NK receptors that we are using. So with the same CAR-T, potentially you can target every tumor. Now we need to demonstrate this in a clinic. However, we have today a very strong preclinical data on many preclinical models. We know that those preclinical models have a certain predictivity, but not 100%. Now we are in a clinic and we are targeting other diseases than CLL and ALL definitely.
Right, so that's a real sort of step forward, that you can take the CAR-T technology and address solid tumours, which is where the majority of issues are with treating cancer. And you said you're in the clinic.
Yep.
What stage are you at right now?
So our lead CAR-T cell called NKR-T has completed the first phase 1 safety study. This is a dose escalation, low dose of NKR-T, because this is the first time those receptors are used in clinic. So we have shown safety profile, a very great safety profile in this first clinical trial. Now we are enrolling in a follow-up phase 1b clinical trial where we are further increasing the dose of NKR-T. And instead of infusing one time, we are infusing the cells 3 times. And we are also enrolling liquid tumors as well as solid tumors. So in total, we are targeting 7 different indications. When we would have completed our dose escalation under this phase 1b, we will expand into those 7 different indications. So the trial will have 7 different expansion arms to confirm safety and also probably to see signs of activity. What we have seen in CAR-T is that the activity of CAR-T is linked to long-time persistence. But this can drive some safety issue because those CAR-T will remain there for a long time, and maybe they will have an off-site, on-target activity that can drive some toxicity. And we've seen in Juno trial that there was some toxicity that were associated with the use of CD19 CAR. So our CAR are not designed to persist and stay into the patient for a long term. So actually they are infused, they trigger their activity, engage the immune system, and disappear. So under preclinical models, we've established that 3 infusions of our CAR NKR-T gives the highest efficacy. So now we are taking this into clinic, and with the idea that this CAR will come, execute its function, and then disappear. Right. So this will result into a better long-term safety profile.
So you do expect to get some kind of efficacy signal out of this escalating dose trial that you're conducting now?
Well, hopefully, yes. I mean, the trial is designed again to show safety, to accumulate as much biological data as we can from the patients. We are also looking to see certain sign of activity, but obviously we would need an additional trial for registration. But this, just will give us a good basis for further development.
And on that's sort of your forward path. I know you've already done one deal with Ono Pharmaceuticals. Yep. Is that the preferred business model, that you will do deals with the bigger players, or do you look to take this through to market yourself?
Our Ono deal is really focused on allogeneic NKR-T and only allogeneic NKR-T in a very specified territory, which is Japan, Korea, and Taiwan. And the reason why is that first, Ono is one of the biggest Japanese pharma focusing on immunotherapy. Ono are the ones that started further developing of PD-1 antibodies 10-15 years back. So it's a great pharmaceutical company that are committed to the field of immunotherapy, leader in their space and in their territory. And Japan for us is not a strategic territory, so that's why this was a perfect fit for us as a first licensing transaction. Today we are not looking necessarily to do any other licensing prior to completing or at least demonstrating some proof of concept in human. And then depending on the data, We will reevaluate our strategy from licensing perspective. If we demonstrate activity and a proof of concept in multiple indication, well, will definitely give us multiple options to do different deals with the same product. If it's only demonstrate proof of concept in limited indications, then we need to consider again whether we want to keep certain indication for ourselves, license others. So all this will be definitely reevaluated based on clinical data.
So as with all these cases, it'll be data driven, but if it pans out, you will have a number of choices open to you there on how you can take both these and your company forward. Well, we'll look forward to hearing more news as it comes out in the coming months. George, thank you for joining us today.
My pleasure. Thank you very much.
For more information about PharmaVentures, visit our website